Hello, my name is Taylor Sugg. Along with Hannah and Lorena, we now bring you the Phylogenetic Tree. Now, the classification basics. You have domain, kingdom, phylum, class, order, family, genus, species. Domain being the most broad, species being the most narrow, meaning organisms in the same species are also in the same genus, family, and so forth. And on the human level, domain is Eukarya, kingdom, Animalia, phylum, Chordata, class, Mammalia, order is Primate, family, Hominidae, the genus, Homo, and species, Sapien. Okay? Okay. Well, the cladogram. A branching diagram showing the cladistic relationship between a number of species. On a cladogram, the simplest organisms are on the bottom, and the most complex on the top. To determine which organisms have which traits, you look and see which notches are behind the organism. For example, on this cladogram, the salamander has all the traits behind it, which is 4 walking legs, jaws, and a vertebral column. Organisms that are close together are closely related. For example, tuna is more related to a salamander than it is a leopard. The notches on the cladogram represent traits the organism possesses. Turtle has all of these, so forth. The phylogenetic tree, which is a cladogram that represents evolutionary change. Basically, same thing, different form. Phylogenetic tree versus cladogram. Cladograms show branching order, although branch lengths are meaningless. Phylogenetic trees show branch order and branch lengths, which means the longer branch, longer length of time. Phylogenetic tree. 2 types of data used to create phylogenetic trees. Shared derived characters, physical traits that represent evolutionary history, which are homologous structures. For example, the non-mammalian vertebrates in the mammals. Hair separates these 2 columns. On the other hand, DNA protein sequence data. Differences in sequences accumulate as species evolve away from each other. For example, you see the human sequence and the chimp sequence, and you see the differences in these sequences. How to construct phylogenetic trees. 1. Determine similarities among organisms. 2. Arrange organisms in a tree diagram showing simplest possible evolution. So, when constructing a phylogenetic tree, it is important to use maximum parsimony. All else being equal, a trait is assumed to evolve once and be present in all descendants. This is important to use because it makes the tree easier to read and it uses less mutations. Sample 1. Create a character table. Well, as you'll see, the chimpanzee has all of the traits. Therefore, the lamprey at the bottom having none. So this is the simplest character down here, and this is the most diverse and complex, being the chimpanzee, and all else falls in between, having a common, Sample 2. Create a tree structure with 5 branches. Well, this one down here is, okay, I don't say that. Okay, I already said that. I'm sorry. Step 3, label the branches. Okay, most complex at the top, simplest at the bottom. Step 4, label and insert notches where organisms evolve, which obviously these are the notches. And you can also must refer back to your character chart to see which organisms have which characteristics. Label the characteristics behind the organisms that first possesses it. For example, in our spinal column comes behind fish because everything after fish has a spinal column. Okay, what not to do? You should not do this. It is complicated and it involves so many mutations because it doesn't use maximum parsimony. So you see, it's just a mess. I don't like it. You don't like it, nobody likes it. Okay? No, we don't. Get out of here. Thank you for your time and cooperation. We love you.
I really hope she can't hear you say